Well, hello, my name is Dr. James Johansson, and I'd like to give a discussion about some of my research. And the title of this presentation is Bacteria Chemoreceptors, Classification and Comparative Analysis and Chemotaxis Control. Hopefully, by the end of our time together, this will make a little bit more sense what I'm trying to convey, but I'll try and unpack even that title here. So what I'll do here is the outline, and just to give the bottom line up front, I'm going to give an executive summary. It'll highlight some of the key things that I've did in my exploration, and hopefully that'll give you a sense as we'll work through it in more details. It'll be a first time through and help you to assimilate the material. I'll give an introduction, talk about what chemoreceptors are, and give a little perspective of what chemotaxis is. And I'm also going to refer to one paper that I found that was kind of interesting, that was about the same time as a predecessor paper came out that I developed. And it's titled Design Patterns of Biological Cells. So you'll see that in a little bit. In terms of the bacteria explored, I'm going to use a paper that discussed 8 bacteria, and then it unpacked the 41 different chemoreceptors that are in those 8 bacteria. And so we'll get a chance to look at that. And we'll also look at the relationship between these sensors, the chemoreceptors, and what they actually sense, the effectors. And so chemoreceptors to effector categories, some of the information I did with the research of trying to get some understanding of these bacteria. So I'll talk about what they do, some of the things that the chemoreceptors do as a part of that. So what the bacteria do and how the chemoreceptors play into that. Then I'll get into detail about a chemoreceptor assessment. And so hopefully you'll find that interesting as we get a chance to get into some of the details here. And I'll introduce some terms that we'll see in just a minute in the executive summary as a first encounter. I'll then get into the relational patterns that are explored. And so here are some of the things. We'll look at the sequence assessments of 3 graph cases. And so basically what I'm going to do is look at the relationship between the chemoreceptors and what they sense. And so we have a lot of graphs that will give us some understanding of what that is. And then I'll look at the sequences, something called bioinformatics, if that's something that you are familiar with. And then I'll start to get into more specifics about some subsuite sequences and what we can see from that. I'll also look at whole sequence comparisons. And there's a couple of tools that help to give us a sense, and I'll compare 2 by 2 for 3 particular cases. In addition to that, I also will look at some of the available annotations for some of the areas for these sequences, for these proteins, and also do an alpha fold comparison, looking at how the folding structure compares. And finally, I'll look at the ligand bonding domain. And this is where, for the chemoreceptors, they actually bond with chemicals, and this is where they actually do that bonding. And finally, I'll talk about, give a summary, conclusions, and then talk about next steps. So let's first of all, I'll just have a dozen charts for an executive summary. And this will be a first encounter for some of these things. And there are some of these areas that I haven't talked about yet, but I wanted to give at least a perspective what I'm trying to do at a top level. And so some of the individuals that I've shared this material, these are the kind of terms that they came up with. This is basically a chemotaxis topology, looking how it's done. And also it has a potential to be doing, quote, structural prediction. And I'll let you be an individual that can be giving some perspective if you think it's going in that direction or not. So Ortega is a key paper that I'm going to be referring to, and I'll be showing that in half a dozen or so slides. But first, let me do this executive summary. So there's 8 bacteria, and there's 41 chemoreceptors. And so this is what I did. First of all, looking at the functional assessment and comparison of these 8 bacteria. Then there's these different ways that how you can be comparing chemoreceptors. They're paralog and orthologs, and I'll do that, and I'll explain those terms in just a minute, but just for now, just say that's another form of comparison. I also did whole and partial protein sequence comparisons, looked at the folding structure, looked at where the chemoreceptors bond, and that part of these chemoreceptors is called the ligand bonding domain. And finally, I'll get a chance to introduce a little bit of what I'm trying to do in the area bonding. So this is a predecessor paper that I leveraged, and it was looking at bacterial chemotaxis control process analysis with SysML. SysML is a modeling language that's used in system engineering. And so what I'm trying to do is to postulate relations that you could observe in the data, as well as confirm or deny things that may not have proven to be correct. And so it's an iterative process and also trying to clarify design element relationships. And for this paper, I introduced this method, the reverse engineering object-oriented system engineering method, or RE-OOSEM. And so for this paper, I'm really focusing on these first three steps. And so you can keep that in perspective. Well, with these 8 different bacteria that I got from Ortega, I started to look at, well, what are the different types of functions that it does? And so I picked out a handful, if it's good or bad. And so this is sort of a way of thinking about that. Is it pathogenic? And so this is just another way of looking at these. Another area I thought was fascinating, and I'll talk about this in more details, biofilms, or I like to think of them as shields. And then finally, bioremediation. Are they actually good at repairing or converting things in its environment into something that's more useful and cooperative for other types of plants and animals? So these are the 8 different bacteria, and I have this kind of rough good guy, bad guy comparison. And so it's roughly even, balance between good guys and bad guys, if you will. But I also want to be thinking in terms of this system engineering type of perspective of what's called a black box assessment. So you may not know how everything works inside, but you can be looking at what are the inputs and what are the outputs, and so you can characterize that even though you don't exactly know how everything works. And I think this is a good analogy for what we're doing with this kind of area of trying to do the first handful of steps for this RE-OOSEM method that I had mentioned to you. Okay, well, jumping forward, I use these terms, and now I'll give a partial definition of what I mean. And so there's something called paralogs, in which it means that there are differences, but there is some commonality in form. And so what I actually am postulating that there's 8 different groupings of paralogs within the 51 chemoreceptors that I'm going to be looking at. And so what I highlighted in red are trying to give you examples of those paralogs that exist for each one of these 8 bacteria. And here are the 8 bacteria. Here's the 51 chemoreceptors. And then for orthologs, it means that they're similar. And so different bacteria actually have the same chemoreceptor. And so, for example, air is seen in this first one as well as one down here. And there's a couple other examples. And 6 of the 8 actually fit into these paralog groups. And then there's 2 other ones, tar and TSR, that are also have these orthologs that show up. Another thing that I did was trying to look at the bioinformatic comparisons. And so a tool that I was introduced to is called OliView. There's a variety of them. And so this was one of the areas that started to capture my attention, that there was almost a exact set of ordering of the nucleotides here, that we can see. All the amino acids are fairly closely related between these different types of bacteria. So that was something that I did. And using those same 8, I also wanted to be looking at the folding structure. And so this is using the AlphaFold server. I can just play this one, and I'll have some more examples later. But if you were to look at those structure, they're basically their folding structure, there's just about identical to one another. So moving on, another thing that I looked at was the ligand bonding domains. And I showed you this chart before. Here are the 51 chemoreceptors that we looked at, and where they are in each one of these 8 bacteria. Some of them have a wide variety of them in the same type of paralog group. But now we have these bonding domains, and there's just a little more than a half a dozen of those that you can see, and I tried to colorize them to make it a little bit easier to see them. And so if I wanted to gather together the information that I've shown for most of the criteria ways to be looking at this comparison, now I'm showing the paralogs, the orthologs, the ligand bonding domains, in which one of these bacteria actually have these chemoreceptors. So there's a lot of information here, and I'm going to come back to this, but this gives us an interesting way to think of where we want to look at some of these and compare them with one another. And so that's something I'm going to continue to do. And for this paper, I'm also going to show another approach of how I could start to identify things that I think would be worthwhile looking at. Another thing that I wanted to include was having a chance to be looking at the modeling ways that we can go about it. And so this is using something in the system modeling language called an activity diagram. And so there's 3 lanes, the chemoreceptors, which we're focusing on here, the chemotaxis, which my last paper I focused on in more detail, and then what is it, the end result is that used by, and so for bacteria, it's motility or dealing with the flagella that may be associated with that. And I also put together something that was artificial. Maybe there's a structure that we see here that could actually be beneficial for us to be using, and that gets into the area of biomimicry, and I'll get a chance to talk about that in a little bit more detail in a few minutes. Okay, so that's just a quick executive summary to point out some of the highlights. And now I'm going to go through the rest of the details and walk through this a little bit slower. Maybe that context will help you. So I already went through the outline. I gave you the executive summary. Now I'm going to get into the introduction, then explore the bacteria, do a chemoreceptor assessment, which you'll see this is a build of these charts that I was showing here on this chart. And so we'll get a chance to be seeing a little bit more perspective on that. And then looking at various ways to get into the relational patterns, and finally talk about a summary, conclusion, and next steps. So some of you've been listening and maybe you don't know what chemoreceptors are, and you can be thinking of these as these are the things that are sensing the environment, sensing different specific chemicals. And so that's what they are focused on. So they detect changes in the organism and the environment, and they play a crucial role in the processes like taste and smell, things that we are familiar with. And so maybe you might want to use an analogy. These are like taste buds, and I'll give some perspectives about that in a little bit. So how are chemoreceptors used in bacteria? They're used for motility and environmental sensing, so it plays a crucial role into that. Ortega, and this is the first time I've referred to this paper, reviews the chemoreceptor systems in classical model organisms and other selected bacteria. And these are beneficial and essential for bacterial navigation. And their ligand bonding domains, the LBDs, exhibit diverse structures and mechanism for recognizing various environmental signals. And I try to have a little bit of a introduction for that in the beginning, and we'll get into that more as we move along. So what is the function of the chemoreceptors? They bond with specific chemicals and aspects of food and toxin sources would be one of the areas that they do, and also sensing the environment. So is it good stuff? That's good information for it to know about. If it's bad stuff, it wants to get away from bad stuff, move towards good stuff. And it also wants to know what's it like out there. And so environmental sensing. So that's maybe a simple way to be thinking about what we're going to talk about. So basically, we're going to have a mixture of engineering and biology. And so I'm trying to show that in a continuum. Biomimicry is where you're doing a lot of engineering and taking some biology and see how you can adapt it. Maybe on the other end, it's more where there's a lot of biology and maybe just a little bit of engineering, where we get a chance to be focusing on bioengineering and systems biology. And so we're somewhere in the middle that we're going to get a chance to maybe, for the most part, look on both sides of those about the same level of detail. Well, let me just quickly use human sensing modalities to make a point about what chemoreceptors do. And I'll just talk about 4 of the 5 sensors. I'm not going to talk about touch. And so human eyesight, we have 2 of them. It gives us a chance to have depth perception. Things that are seen with the eyes are then focused onto the retina, and that information is taken in the optic nerve to the brain. And so the brain is a key element in that overall processing. It's one of the most complicated sensor that we have. Hearing, once again, we have 2. And so there's a modification of what we get a chance to be hearing with our ears, amplified, converted, and that is brought to our brain, and we can get location perception because we have ears on both sides of our head. Smell, this is a little closer to chemoreceptors, and so we can detect odor molecules. There's the olfactory receptors, and then there's signal transmission and that is brought to the brain. Maybe you can get a chance of you can locate smells. If you've ever seen maybe some of your pets as they're just sniffing and they kind of sniff and they'll move around and sniff in a little different direction. They're trying to get a chance to sense gradients, which is the direction in which it's stronger. And that gets us to, that's one of the things that is definitely something that's going on in chemoreceptors. And so somewhere between taste and smell is what we're talking about for chemoreceptors. And so for the taste buds, there's 5 basic taste. And so maybe you could say that we have 5 different types of chemoreceptors in our taste buds. There's signal transmissions, there's taste and other senses that we get a chance to be seen in, and there's also brain processing that's part of it. Well, actually, one of the recommendations that was associated after my previous paper was published was a paper titled Design Patterns of Biological Cells. I'm not going to spend too much time on it, but it is actually an interesting paper that I think is relevant in terms of starting to look at what you would be, from this case, I'm looking at design patterns in software engineering and computer science and apply it towards biological cells. So they had to come up with these different kinds of classifications. And so the 3 classifications that came up were creational, structural, and behavioral, and they have the details for each one of those. So, and they also, so this is the creational. For the structural, here is a listing of what they have. And I put a star by ones that are relevant for things that I'm going to get a chance to talk about a little bit in this discussion, and input and output is one of those. And then for behavioral, we have the adaptation and switching. Those are things that we definitely see. Well, in terms of what we're going to look at for the bacteria that are being explored, I already mentioned Ortega. This is the paper that was extremely useful. It was a survey paper, and it gave us a chance to be looking in more details of what these different type of bacteria do. So the chemoreceptors in bacteria detect a variety of signals and feed that information in the chemosensory pathways that represent a major mode of signal transduction. So this is just the very beginning of the whole chemotaxis process. Chemoreceptors differ in topology, sensing mode, cellular location, and above all, the type of ligand bonding domain. And so all of these types of factors are there. And although signals can stimulate chemoreceptors in a variety of different ways, diverse ligand bonding domains appear to employ universal transmembrane signaling mechanisms. So I just highlighted a couple things from the abstract from that paper, and I'm going to be using these 8 to help me in my characterization. And so this is, I'm just going to be using the abbreviations. So if you hear me call one of them out, this is what I'll be referring to, and either a 2 or 3 letter shorthand way of thinking about them. So, and I already introduced this idea of a black box assessment. We're not going to know how everything works inside, but by getting it from a top-down, object-oriented perspective, this is how we can start to explore and hopefully uncover more details about how they work. So the first one, E. coli, it has helpful and harmful strains. It's something that we want to live in our gut, the good strains, and it's actually used quite extensively for a lot of different forms of experimentation. So Salmonella, it's a harmful pathogen. It's used for a model of understanding pathogenesis, and it can invade host cells and evade the immune systems, and so has the ability to be nefarious. And so unfortunately, this is definitely a bad guy. Bacillus, found in vegetation and soil. It's forms of protective biofilms, and so it has this shield that I think is interesting. It's used in research to study gene regulation, signal transduction, and cellular differentiation. And it's used in a variety of different areas, production of enzymes, antibiotics, and other biomolecules. HP, it causes ulcers, it neutralizes stomach acids, just to mention a few things that it does. And so causing ulcers is not a good thing, so I put it in the bad guy category. CJ causes foodborne illnesses. It's found in raw or undercooked poultry, unpasteurized milk, and contaminated water. Not a good guy, and so we have another example of that. PA, it's known for its ability to survive in diverse environments. It produces toxins to damage host tissues. Once again, puts it in the bad guy category. But it also has this interesting thing, biofilms, shields, to help resist antibodies and immune response. PP, it plays a role in breaking down organic matter, and so it's a good guy. It's used in production of a variety of things in various circles, bioplastics, various other compounds. It's capable of degrading a wide range of pollutants, and so really wonderful that that's part of what we have. In PF, it contributes to the nutrient cycle and organic matter decomposition. It's non-pathogenic to humans. So once again, I would put that in the good guy category. And so this was a synopsis of that. These first two are tied up with this good versus bad, but I'm being more specific in terms of saying that it causes pathology. Here, biofilms, about half of them uses shields, and about half of them does bioremediation. So remember, E. coli has good strands and bad strands, and so that's why I introduced this yellow in the middle to give us a perspective of that. Well, okay, well, the chemoreceptors sense the effectors. These are terms that Ortega uses. And so I'm going to be using those same terms. And I found it interesting that there was actually a two-way street. And so the bacteria is wanting to sense the environment, and in more of an engineering term is doing surveillance. And so this is moving from the environment to the bacteria. But there's also situations where it goes the other in a direction, where the bacteria is trying to adapt the environment. So it could be mitigation, countermeasures, and spoofing. And so once again, these are engineering terms that I'm using just to give a perspective of what you can see in these various types of bacteria and what the chemoreceptors and other parts of the bacteria are involved with. So I did some research, and so here are some of the categories that are associated with each one of these bacteria. So for E. coli, there's virulence, and so that's related to the bad strands, substrands, metabolic communication, antimicrobial, and host modifying. And so since this, I'm still putting E. coli in the good category, good guy category, although there definitely are some that are not. And so we're seeing a mixture of these different kinds of effectors and the things that are going on. For Salmonella, invasion, immune modulation, intercellular survival, inflammatory response, and metabolic. These are the types of categories that start to come up with Salmonella. Once again, this is a bad guy category for that individual bacteria. Bacillus, it's antimicrobial, it has biofilms, signaling, enzymatic and host interaction. So, but overall, not doing too much in the way of nefarious. And so I don't see it as bad as some of the others that we've seen. HP, virulent, immune modulation, adhesion, metabolic and inflammatory. And so it's trying to attach and start to change what the host area is doing. So definitely not a good guy. And so we can be seeing some of the ways that the effectors and other parts of the bacteria are being used. CJ, it has adhesion, invasion, immune evasion, toxin, and metabolic. And so this is definitely something I would say would be on the nefarious type of category. PA, virulent, biofilms, toxins, and immune evasion, and metabolic. So those are some of the things associated with this one, and I would still put this in the bad guy category. TP, I would put in the good guy category. Bio degeneration, biofilm, stress response, metabolic and plant interaction. It's trying to actually improve things around it, take care of toxins or things that need to be mitigated. And so definitely is a really a good in those types of areas. And finally, for PF, it's antimicrobial, biofilms, root colonization, induce systemic resistance, and metabolic. And so it does a variety of different things. And so this is just trying to show that all in one slide. And here is just trying to show it with a colorization of either the good guy or the bad guy. And then I started to try and look at this. What are some of the categories that you might infer from all that? Things related to metabolic activity is pretty high. Half of them are dealing with biofilms, or I like to call that shields. And then I looked at the different categories. There's a wide variety, but metabolic and biofilms definitely show up as being large. And then if I wanted to take it up a little bit level, because this is a lot of categories, I then massaged it and came up with these names. And so if I was to put that in a pie chart, this is a way of looking at that the top level one would be protection. Doing the bioremediation breakdown would be a large one, looking for food sources. And so that's just another way of thinking about it. And this was just another form of looking at that for each of these 8 bacteria. Okay, that's just a lot of information, but it's useful for a way of starting to figure out, well, what are the characteristics of these different types of bacteria and characteristics of what the chemoreceptors do. Okay, in this section, I'm going to have a deeper dive to look at the chemoreceptors. Going back to Ortega, this was quite helpful for me since it's a survey paper. It lists all of the different chemoreceptors associated with each of these bacteria. And so if I was to count that up, and there are situations where some of the chemoreceptors are in more than one bacteria, you might remember the term. It's called an ortholog if that is what is there. And so this is just looking at put them all in alphabetical order. And this was a helpful way for me to get a chance to be thinking about the paralogs and getting a quick visual perspective of which bacteria has what. So there's a total of 51. And so, and then colorizing the paralogs, this is the things that I have there. So there's 8 of them, but let's take now a little bit more of a detailed perspective of looking at, well, what are these two different things, the orthologs versus the paralogs? And so ortho and para are co-opted from chemical terms. Ortho, you can think of it as adjacent or maybe a specialization. And so they have a similar function. So tar, that's in more than one bacteria, and that's one of the ones I'll be looking at, that they're orthologs. We'll get a chance to be looking at that in further detail. Para, on the other hand, is opposite, or it has a different function. And so, but maybe there is some structure connection related to what we're seeing in that. And so that's something that also useful. So I'm postulating these 8 paralog groups. And so I'll just briefly try and give a description of what the commonality that is among them. So air, and there's 3 of those, energy taxus that detects oxygen. CET is also a energy taxus, and it gives us the bacteria the insight to be able to move towards or away energy sources of one or another. CTA, it targets amino acid groups, anywhere from a wide range of amino acids to more focused. And so that's what we see in that paralog group. CTP, it senses inorganic phosphate and moves toward or away it. And there's another one in that paralog group that senses malate. There is the MCF, it detects the response to acids, TCA cycle intermediates like the ones that are shown here. MCP are methyl accepting chemotaxis proteins. And when I see methyl accepting, this to me is one of the things it implies is that this shows adaptation. You can have the methyl group actually increases or decreases this functionality, and it makes me think about epigenetics, how the epoch actually tempers or enhances what's going on for what's encoded with the genetics there. PCT detects specific amino acids, and this the focuses on the L amino groups or a couple other things here. And so basically it's not just generic amino acids, but a certain ones. And then finally, TLP, it detects acids, urea, lactate. And so a couple of specific types of chemicals that are captured in that group. Okay, so here I'm listing those 8 groups, and I've numbered them 1 to 8. And so that gives us a little bit of insight. But there's also some orthologs, and so TSR and TAR ones, and I'm going to be focusing a little bit later on tar, and so we'll see how closely those compare. Well, now I wanted to start to build up the bacterial paralogs, and we can see how many paralogs exist in these different types of bacteria. And so I'm listing the bacteria over here and the groups. And another way that might be helpful, and this is what I showed earlier, was I just highlighted in red how we see that. So we see the bacteria are listed going east and west here, and it's the flip of what's over here, but this is just showing the groups rather than individuals. So it's a slightly different perspective of looking at for both of those. Then for the orthologs, we can see that the orthologs are only being shown in two of these groups, the air and the MCP. There's also a couple that aren't fitting into one of those paralog groups, and so I also wanted to highlight those. And so the TAR and the TSR are not in a paralog group, but the other six are, and so that gives us a little way of capturing that. So just wanted to come back to this chart briefly because I'm going to colorize part of this chart with this good guy, bad guy thing here, and we're seeing it over here. And so now this is trying to show the relationship between the good guys, and so all the ones that are in green. And we see that showing up here in this MCP group. So that's for the good guy that we see some interesting areas that we could explore, things that go across that paralog group. For pathogenic, so I'm using a different ones from this group here. This is the second one over there. And if I was to sorry, my mouse is bouncing around. So pathogenic, we have this. And then if I start to pick it up, so the ones that are green, the ones we'd be focusing on. So if we're trying to look at some ones that would be how they might use different paralogs, this just gives us a couple examples. Biofilms, I always like this one. And if any of you are Star Trek fans, this would be maybe something that you're familiar with. And I think of biofilms as like the Star Trek Enterprise putting on its shields. And so these are the ones that would be applying there. So 4 of them actually have biofilms. And what is common between all of those? Well, actually, there isn't one. But maybe if we started to look at some of the other aspects of it, including the ligand bonding, the folding structure, stuff like that, we might get some additional insight. And finally, we have the bioremediation. So we have these ones that we can be looking at for those possibilities. Okay, so that's a lot of information. Hopefully that is helpful for you as we're trying to unpack this. Another thing that Ortega annotated was all of the different effectors. And so this is an approximate list that capturing for what he has in tables, the text. And so there's on the order of about 70 different effectors that he's captured in his survey article that the 51 chemoreceptors are going to bond with or have a response to. Okay, this is going to be the first of 2 times I'm going to use this. What's an engineering impressionist? So I'm trying to apply engineering towards a biological system. And so what I would tend to think is the more chemoreceptor types used by a bacteria should correlate with finer, more precise sensing and characterization of the surrounding environment and serve a useful purpose. So that's my thought process here. And what I wanted to use was graph theory as one of the ways maybe I could start to be looking at what are the characteristics that we're seeing in these 8 bacteria, the relationships between the chemoreceptors and the effectors. So one at a time, I'm going to go through each one of these bacteria. E. coli, it has 5 chemoreceptors and it has a handful of effectors. And so I just started to annotate a few in terms of what they are, but that just gives you a little bit of a sense. And so first for E. coli, Salmonella has about the same order, not nearly as many effectors, but a handful. BST, 4 chemoreceptors, 5 effectors. So, and you can see some of these are trying to look at a subset of amino acids. Some are a little bit broader. We can see that being played out. HP, so it has just a handful of chemoreceptors, but it actually has a lot of effectors that it's sensing. And so some of these can sense quite a lot of different individual types of chemicals. CJ, there's a large complement of chemoreceptors. Once again, some of them are sensing quite a lot of different effectors. And then we have PA. There's so many that I have to straddle over a couple of slides. So this is the first of 2 slides, and here is the second one. And so we can see this is actually fairly complicated, and thus my abductive argument that, well, there should be a lot more functionality in these that are having all these different characteristics. PP, once again, it requires 2 pages to cover it. So this is the first page, and this is the second page. And then finally, the PF, kind of a modest in comparison, more like E. coli and Bacilla, the very first 2 that we got a chance to look at. Okay, well, now that we've got those graph theories, I'm going to take that as a sounding board. This is one of the options I could be looking at for trying to uncover relationships. And so there's 3 different relationships that I'm going to explore. So I'm going to explore a particular chemoreceptor and what it senses, that is the effectors, and that should suggest function and how it supports the observed behavior of the bacteria. So the first one is looking at these 2, and these are orthologs. And so they're both have tar. And what do they actually sense for? Well, they do sense one thing, aspartate. They both sense that, but the other 2 are different. But what can we observe in the structure, the amino acid structure, and the folding for each one of these? We'll get a chance to look at another one we'll look at are the comparison of PF and PP. They're different chemoreceptors, but same effectors, but they still fall into the same paralog group. And so that will be the second one that we'll look at. And finally, we'll look at multi-sensing chemoreceptors. And so there's enough of them. I took 2 slides to put it on. So first for CJ, this is an example of one of its chemoreceptors, and it senses for a lot of different things. And then for HP, it does something similar, senses a lot of other things, but these are not in the same paralog group. And so we'll think about that. Another one that I did not look at in as much detail, but I'll just have one slide here. This is a build that I can be thinking about, well, what is it telling us? And so here we see 5 different chemoreceptors sense the same type of effector. And so is there some commonality that I can observe that's going on here for this bacteria that I've highlighted in purple here? And so these are the 5 different chemoreceptors. And I did notice that 4 of the 5 actually has the same ligand bonding domain. And this other one, the way it's listed is none, but I wonder if that means that it just hasn't been figured out yet. So maybe that might be one of the things that would be interesting to think about. So for the sake of what I'm going to focus on, I found 3 different cases that I'm going to compare 2 by 2 for this part of my presentation. So the E. coli and Salmonella, look at those on PP and PF. And so these are orthologs, these are paralogs, and these are just showing both 2 complicated types of chemoreceptors that sense a lot of different things, and can we observe anything about their structure? And so just to use that one chart to show those 3 groupings, this just might be another way to think about it. So here is the ortholog. Here is, anyway, these are the 3 groups that I wanted to get a chance to have us to refer to, okay? Okay, let's now get into the sequences. So for the bioinformatic assessment, this is where we look specifically at the amino acids. And so here, if I look in UniProt, these are the details of what shows up. And these are actually short enough, I could actually show the full sequence. And then I can do a comparison. So this is one of the tools, Cluster Omega, to compare them one to another. And it turns out that they're about 79% the same, but that means 21% different. From an engineer perspective, that just kind of strikes you a little bit funny. And I'm just going through these slides to highlight the comparison between those. So that's the first of 3. This is the second one. Now we have 2 chemoreceptors that are sensing the same effectors. Here are the UniProt proteins that I looked at. Here's what it looks like in cluster omega. And then I zoom in to try and make it a little bit easier to capture that. And so there is some commonality. If you would see a bunch of stars down the bottom, that means that they're the same. So just scanning through that, here we see one section where there is some commonality between those, and then I've annotated that here, and I'm going to come back to that in a little bit. So with that one sequence, I used BLAST to try and find out, well, what are some examples of code snippets, amino acid snippets that would have that same characteristic. And so here were a couple, and I'm just going to focus on these. And so methyl accepting chemotaxis protein fragment, methyl accepting transducer domain containing protein. So these are fragments that are focusing on the methyl accepting transducer. Interesting thing for us to be thinking about here. And I'm going to be coming back to that again in a little bit. Okay, here's multi-sensing, the 2 cases. Here's the UniProt proteins. And then I looked at the sequence here. Now, once again, I'm trying to see where there is a commonality. I'm going to zoom in, but where you're going to see it's just this area where there's that commonality. So just going forward, handful of slides, there's the commonality. There's the listing of what those amino acids are. I looked at in BLAST and found that these are the top ones that come up. And I'm just going to be focusing on this one. And once again, this is a methyl accepting chemotaxis protein fragment. So then when I started to look at them closer, it turns out that these are focusing on the same area. The same effectors actually has some commonality of a few more amino acids, but those are pretty much the same thing. Okay, another perspective that I looked at, and I'm going to come back to what I just did with this whole sequence comparison. And so now, still using these same 6 areas that I've looked at already, I'm going to be looking at these 6 different chemoreceptors. And so the tool that I'm using is AviView from Uppsala University. And so first of all, I'm going to give a view that's a top level. So this is like really impossible to read. This is a little bit better. And so let me go ahead and show you the areas that I'm going to focus on in more detail, where I could be starting to look at. So in the executive summary, I focused on this area where there's commonality across all 6 of these chemoreceptors. There's places where only 2 have distinctive features and not the rest of them. And so I'm going to highlight those and dwell down on those to look at their structures. I'm not going to use this one. This is a way of looking at differences only, just trying to highlight that, but I'm just going to refer to the other one. So this is what it starts out the sequences. I didn't use that one, but here is the all similar region. And so I'm annotating it there and then had it for what we see for all 6 of the specific chemoreceptors. That gives us an idea. And this was interesting that I had looked at this ones for the same effectors and the multisensing, and those all highlight the same common area. So kind of stumbling on the same information from a couple different points of view. So these are the methyl-accepting chemotaxis protein and the methyl-accepting transducer domain containing protein, both of those being fragments. And I just highlighted that there so you can see it a little bit better. Well, for the TAR, for E. coli and Salmonella, these are a section that has some degree of commonality, but I would argue not as high as I would like, at least from my engineering mindset. And so that just gives us a chance to be thinking about that. Then for the similar same effector subsequence, we have those. It's only about 28% the same. And we can see the differences between those. And then just looking at it a little bit more for the quote similar PF and the CJ portions that were highlighted, you can get a chance to look at those sequences. And those are things that I'll be coming back to in a minute. So we looked at these are kind of building on the same area. We started with these 6 different chemoreceptors, found some areas that looked interesting, are going in a little bit detailed. So now we're going to still use those same 6 chemoreceptors. And first, there are various websites like this where you can get some interesting annotations, and when you're getting into focusing on different parts of the protein and see what does it refer to. So these are just 3 of the 6 that actually showed up. So I have it for both of the TAR cases and this other one, CCML, but I didn't have it for all 6. So I'm just going to leave that and now just focus on this other thing with this AlphaFold server to get a chance to be looking at the structure. And so here's the first 2. You can see that, well, if I rotate this and I look at how their structure is, there actually is some fair amount of similarity between these 2. So these are the TAR. Towards the end of the sequence, those are structures that are common. Now we have the same effector, and so there's a place where they had some common structure here. And so now if I just show you what the AlphaFold server shows for that one, this just gives you an idea. Once again, they're pretty similar. Maybe one has a little bit more of a bend, but this is only 28.6% commonality, and yet we see this, the folding structure is fairly similar. So it just goes to show you, just having the same sequence is just one of the variables that you can be using to figure some of this stuff out. Next, we have the similar PF and CL, the things that showed up that I didn't expect to have some similarity, but let's look at what we find for that from a folding point of view. And once again, these are really, really close to the same type of structure. So once again, interesting to have us consider those both portions. And so these are another example where we can be looking at the comparison of their structure. So some similarity, but I would say not as much the same as we're seeing from the other cases. And then finally, for using looking at the folding, here's a case where I'm showing all 6 of them that has this common area. And this is actually, you really do see why it would come across that these are all similar, because they all have the same general types of look and folding that they exhibit. All right, now we're down to the last portion of looking at this comparative analysis of these bacteria, and we're trying to pick out a handful of these chemoreceptors to see what we can look at. So I've shown you this chart that I used for helping us to identify paralogs and orthologs up here. But now what I've done is I've brought this part down on the bottom for the ligand bonding domains. And I showed you this in the executive summary. So maybe this is now when we get into it to make a little bit more sense. So here's an example, like all of the air, all of them has the same type of ligand bonding domain. You could argue that that would tend to make sense. Here for the MCP, most of them are the same, using the DCAS LBD. Well, what is the distribution of these different bonding domains? And so this just gives us an idea for this 51 chemoreceptors, 35% or a third of them, a little more than a third, are DCAS. And then it just goes down from there, HBM 15, 4HB 14, past 12. And then there's others, just a catch-all that I didn't focus on in my analysis. And then I just started to annotate a little bit which groupings of the paralogs for the chemoreceptors fall where. Okay, just one last build chart. And so now I'm showing the ligand bonding domains with the orthologs. And then here is the ones with the paralogs. And if I put them together, this just gives us a way of thinking about that all. Although I didn't look at it for this presentation, this would be something that I'd like to explore, the paralogs, and looking at the structure, the coding sequences, other factors for PP. That would be one that I think might be interesting to look at it. But I think there's a lot of other areas that might be helpful. So we covered a lot of ground in this presentation, and so you can always feel free to review this, go back, pause, ponder on this, and you can also reach out and communicate with me that we can have a chance to have a dialogue. So what's the summary of the findings? This assessment compared 8 bacteria highlighted by Ortega using an engineering methodology. RE-OOSEM, you might remember from my executive summary, starting with express functions and looking for related patterns and protein subsequences. We found a common 29 amino acid subsequence for all 8 bacteria that correlates with a methyl accepting transducer domain. Salmonella tar and E. coli tar are 79% identical and have the same number of amino acids. They also contain the 39 amino acid subsequence unique to them, and I showed that by looking at its structure. The quote same effectors for PP and PF using the MCFR and MCPS. Chemoreceptors have a similar amino acid subsequence of 10 out of 35 that are the same, which equates to 28.6. And the PF-MCPS and CJ-CCML have two 27 and 47 amino acid subsequences with some commonality, and I showed the structural relationship for those. So graphing chemoreceptors to effectors for each bacteria shows an alternative way of characterizing their function, their focus and function. So there's variation in the amount of food on focus on food, toxin, communication, protection, attach, and breakdown functions. Some of the things that I've tried to focus on for categories for effectors. The sequences reveal some correlation with observed function. More work could be done in this area. And with some of my build charts at the end, this might be some interesting things to give some more perspectives on what can be thought out and drawn out for things for additional research. Some bacteria are primarily helpful for the host, while others are detrimental, even though common subsequences exist. Some common shared functional elements are found between the good guys and the bad guys in terms of the bacteria and how they're utilizing their chemoreceptors. So more work will better quantify the relationship between observed function and realized design features, looking at other areas explored by Ortega, including the ligand bonding domains in more detail, highlighting the three primarily ones that he talks about. I actually show that there was, for the 51 chemoreceptors I have, that there was fair distribution common between them, using the system modeling language, modeling to explore from another vantage points the similarities and different relationships between the 8 bacteria should help us to get some insight. The more relationships show up that merit reviews, additional orthologs like TSR and MCP paralog groups for PP, which uses 8 of the 12 paralogs. I already mentioned that in just a few slides ago. And another thing that is something that's found on the engineering side is something called an analysis of alternatives, where you get a chance to explore the sensitivity. And so we can be looking at the bacteria when we consider the difference between the requirements, an engineering term for what it does, the design features for the chemoreceptors that are required to meet the objective functions desired. So basically a sensitivity analysis that you could be stating, if I wanted to do something that's going to be good for the gut, the human gut, which one of these 8s is designed that would definitely focus more on that. It's the answer that we see is E. coli, and so we want to figure out what actually aligns it to with all these different factors that are available for us to be setting its function. And then just lastly, talking a little bit more about modeling. And so for the biological world, this is what we're looking at. The chemoreceptors are the beginning, then flows into the chemotaxis, and finally it gets into how you activate the flagellum. It deals with the idea of motion and motility. So this discussion has been on the chemoreceptors. And so when we start to go from the descriptive to the prescriptive and try and go to RE-OOSEM to pull it out and be able to look at things from an engineering point of view more. What does a bacteria do that helps us to get insight into its function? How does it do it? This shows us the design. How many? That gets into parametric type of traits. What kind? And so the analysis of alternatives of how we can be comparing, we know what the complement is for each one of these 8. Which of these 8 would be the most optimal to be doing a certain type of behavior? And then what for? This gets into the requirements. And so if we get to the end and we know exactly what it's done through a lot of good research, things like what Ortega has pointed out, we can definitely see how we can get into a typical engineering mode where we can be requirements driven. So what about having things that are not only in the biological world, but there may be something that could be used for crossover, whether it's molecular machines or maybe it's not even in the biological world, but a simple, low-cost sensing reactionary device that could be helpful. Maybe this could make a more efficient pool cleaning system that maybe you've seen those that for various homes that they're just trying to vacuum all the leaves and stuff on top of the pool. What if you could design it that it can actually ingest some of the chemicals of the sunlight, various sources of energy to self-propel it, and also have it sense where it needs to be spending most of its time, where's the most pollutants that need to be mitigated. And then that gets into starting to think about the models. And I showed you this already, but not only can you be doing modeling, but you can actually do executable models that you can check out logic and flow of things and see if there's anything that you might see that maybe you haven't caught. And so from starting out with a logical perspective, then you can get into things that are at the physical level. All right, well, thank you very much. That's hopefully you enjoy this, and that's going to end this lecture. Bye-bye now.